So our objective today, so let's go ahead and list on our objectives, is 12/10/2018, and our objective today is to, one, finish transcribing and translating, right? Okay, so going over this this transcription translation, at this point you should be able to answer these quickly. What I expect is here, as long as I hear most of you, you know what, I don't want to say your names while I'm recording this, so I might point to you and say, what do you think? And I'm going to expect to hear an answer from you. If I don't, I'll expect to see you during advisory. I'll give you a special pass so you come and see me during advisory. We're going to have to discuss this. Those of you that were absent for whatever reason, you need to know this. We went over it in class several times. You had an assignment. It was, I don't know what else to say. I uploaded the video, I'm told, explaining how this works. For people that come to me today and ask me, what do you do here? I'm like, I told you, why do I have to repeat myself? And I uploaded the video. Go watch the video, all right? Gene provides instructions for making a protein, and proteins influence our characteristics. It's bold-faced, so you think it's important? What do you think? Do you think it's going to be on a test? Do you think it's something you need to know? It's bold-faced? What do you think? Participation, do I have it or not? Yeah. Then let's hear it. What do you think? Do you think it's important? Yes or no? Yes. Yes, it is. Protein. What is a protein? Somebody.
Chain of amino acids.
Chain of amino acids. A bunch of amino acids. They could be the same amino acid or they could be different ones. It all depends. 1 through 20, right? A bunch of amino. It could be a few amino acids. Could be a long chain, whatever it is. What do we call this? The structure. What structure? The order of amino acids. Is the primary structure is correct. Very good. Excellent. I'd like to hear this. All right. Instructions to make a protein provided by the gene, which is a specific segment of a blank. What's a gene? A specific segment of what? DNA. A DNA molecule is correct. What do we call that DNA molecule if it's all wound up?
A chromosome.
A chromosome. So a chromosome is DNA, as you will learn soon enough, that is all wound up. Now you hear this, those of you that maybe you're a little confused, you hear this, these people responding, they know this because we've covered it. So let's make sure we get this done. All right. What do you see? Specific sequence of nucleotides. A gene contains a specific sequence of nucleotides, instructions for a specific sequence of amino acids, right?
Yep.
So you have this code that leads you to a code of an order of amino acids called the primary structure. Determines the structure and function of the enzyme and protein. We talked a little bit about why that does, right? Mm-hmm. Because the different side chains can change the shape of the protein. That shape of the protein is going to change its function. It's no good to have a rope that looks and acts like gelatin, right? Would you trust a rope made of gelatin to save you from falling off a mountain? No, because its structure is not made to hold weight. Both gelatin and and a rope are two versions of a polymer, right? But I wouldn't use gelatin to try to climb down, and I wouldn't use rope I wouldn't eat rope, right? So they're two different structures that have two different functions based on the order of those amino acids. The gene directs the synthesis of protein by a two-step process, right? The first is what? What's the first step? Transcription. That's right, transcription. What does transcription do? What do we do in transcription?
It translates the DNA to RNA.
No, it does not. You said, and listen to what you just said. Listen to what you said. You said transcription, that was correct. And then you said translates. Does transcription translate?
No.
No, it does not. Two different. I know it's tricky, but you just have to learn the vocabulary. Two different processes. So what does transcription do?
Determines the sequence of nucleotides.
No, specifically, what does it do? Somebody describe it to me quickly. I don't have time. What does transcription do? Describe the process. Go.
Transcription turns DNA, nitrogenous bases
Yeah.
into mRNA.
Perfect. Perfect. Great job. Absolutely right. Absolutely right. Transcription, you're taking DNA, nitrogenous bases, deoxyribonucleic acid, nucleotides, right? A sequence of those, and you're transcribing them the same message. You're transcribing the message into RNA. Why are you staring off into space? What's going on in your head? What are you thinking about?
Nothing important.
Okay, so let's focus on the important part. All right. All right, so messenger RNA, mRNA, what does it do? Go ahead. You were daydreaming? Let me know. What does messenger RNA do? Don't look it up. Messenger RNA. What does a messenger do? What does a messenger do?
It sends a message.
Takes a message. What message could there possibly be in all this discussion? Sequence of amino acids. And where it's coming from where? From DNA, which is in the nucleus. To where? The ribosome, which is where? Cytoplasm or rough endoplasmic reticulum. That's right. Good. So we're ready to go then. It sounds like you could probably pass the test right now. Some of you. All right. Transcription, it says here, transcription is a nucleotide sequence in the gene and DNA in the chromosomes, the original message. The molecule that is synthesized is what?
mRNA.
mRNA is ultimately what's and where location where it takes place, nucleus. What about the original message for translation? Where is it?
Nucleotides.
What nucleotide sequence? The original message or instruction in the process of translation. The beginning, where's the message in translation?
mRNA.
mRNA, that's correct. mRNA. The messenger is which molecule synthesized? Protein is correct. Location where it takes place, where does it happen? Cytoplasm. Really easy. Yes? All right. Next, let's take a look. This should be really fast. What is hemoglobin?
A group of 4 proteins in red blood cells bounded by an iron atom.
Can I say what I said?
Yeah.
I said a 4 protein molecule that binds oxygen to your cells.
Not quite to the cells. Binds oxygen says and delivers it. This is the key. It delivers it. Let me just say. When somebody says you're anemic, it's because there's something going on with that process of binding the oxygen and taking it to your cells. The interesting thing about hemoglobin is that when there's high concentration of oxygen, remember we talked a lot about concentration. Why? Because it's incredibly important in biology. When you have a high concentration of oxygen, hemoglobin binds oxygen. When it gets low, when that oxygen concentration is low, it releases oxygen. So in the lung, where there's a lot of air, what do you think the hemoglobin does?
Delivers oxygen.
Wow.
Wait, say it.
Let's try that again. All right, so I don't know if you guys listen to yourselves. So I'll say it. Hemoglobin binds oxygen when oxygen levels are high. Are oxygen levels higher in the lung or in your internal muscle tissues in your toe? In your lung. And you said that like it's common sense.
Yeah.
Okay. All right. So in your lung there's higher concentration of oxygen. So hemoglobin binds oxygen when it's higher concentration. So the hemoglobin is going to do what in the lung? Bind oxygen. All right. It's not going to deliver oxygen. It's going to bind oxygen in the lung. What does the bind mean? It's going to grab it, right? Fix it, whatever you want to call it. It's going to take that oxygen and do what? It's going to get it's in the red blood cell. So where's the red blood cell? Where in the body?
In your intestines, in your.
But where is the red blood cell? Where is the red blood cell?
What do you mean? It could be any.
It could be anywhere, but where is the red blood cell? Where is it? If I were going to cut you open, where would I find a red blood cell? In your blood. Oh my God. I'm asking simple questions because I need you to really process this stuff and really think about what it means. I mean, they're simple questions. The red blood cells are in the blood, right? The oxygen the blood gets close to the space that's in your lungs. It's outside. We call them the alveoli. Those of you that went on the anatomy camp, you saw the lung. It's sitting there right at the edge. The blood vessel touches that space but doesn't cross. The blood cell gets close. Oxygen diffuses, because oxygen is a nonpolar small molecule, so it diffuses easily into the bloodstream. The oxygen gets into the bloodstream. What's the hemoglobin going to do at the lung? It's going to do what? Bind it. It's going to hold on to it. What's the heart doing to that blood cell? Pumping it. Boom, 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 boom. Away goes the blood cell down down the blood vessel to a tissue. Pick a tissue. Pick a part of your body. Muscle tissue where?
In your arm.
In your arm. So there you are, the muscle in your arm. There's the red blood cell. Comes next to a muscle cell. That muscle cell is starving for oxygen. There's not a lot of oxygen. There's a lot of CO2, not a lot of oxygen. What's that hemoglobin going to do now? Release the oxygen. Why did it release the oxygen? Because it delivers. That's right. Because it doesn't hold on to it. Chemically, it does not hold on to the oxygen when there's low oxygen. Are we clear on this? So the oxygen is let loose. What is the cell going to do? Take it. It's going to use it. And what's going to happen to that oxygen? Mitochondria is going to take that oxygen and make what? ATP and water. Right? So oxygen is going to turn into water. Yeah.
So it just disperses anywhere that red blood cells are.
Anywhere red blood, that's why you have red blood cells in your body. And you can see if you have less red blood cells because you're bleeding, for instance during heavy flow of menstruation, or when you're bleeding out because you've been cut or shot or hurt, or if you have problems with your bone marrow, you're not producing enough red blood cells, or if you're diabetic like myself, or if you're obese and you're too big, you have too much blood and it's hard for your bone marrows to keep up with the amount of blood that's needed. If there's all kinds of issues, sickle cell, which is what we're talking about here, right? You can't bind oxygen well because the hemoglobin is not formed well. The structure's wrong, right? So it doesn't hold on to oxygen well. So at that point, all those reasons can make you anemic. Any one of those reasons can make you anemic, which means you can't get enough oxygen to your tissue. You feel weak. Why do you feel weak? Because you can't make what? Without oxygen. You drink, no, we don't. We drink water. We produce it, that's true, but we don't need it. What is the real reason? ATP. ATP. Yeah.
So, is that why, like, when you're, like, doing an exercise, if you're breathing in really hard, is that, like, why you start to sweat because of the hemoglobin?
No, you sweat to cool off.
No, but, like, you said after, like, it produces water.
You're producing water, but it's not really, the water is not, you need to drink water. You can't make enough water to keep yourself hydrated. So we constantly have to drink water, so as humans. Other organisms do a pretty good job at doing that. The sweating happens in order for you to cool off, because you have to keep homeostasis. Remember, you're a system. I keep saying that. There's real deep meaning to that. It's just not a word I'm throwing out there. You are a system. So there's all kinds of things all happening at once. It's layers upon layers. It's like a web. You pull on one string, the whole web vibrates. All right. So there's all kinds of things that are happening. Very few things are very simple. They are all interconnected, however. All right. So let's move on. So simplifying those nitrogenous bases and ribose sugars and phosphates that we drew up, right? Remember us drawing all that, right? Simplifying it to just 3, and in fact we did simplify it as well. I think we put NB and we put a little square, and then we said ribose sugar, and we drew a pentagon, and then we drew a phosphate. We do something like that, right? And we said that's not the correct placing, and actually this is wrong too. So there it is. Right? You remember this? We did this long ago when we went over in fact, I'm gonna print out and go over again some of these structures that we'd gone over. We have this long chain of DNA or RNA, right? And then DNA, there's double-stranded, and they form a double helix, and then the hydrogen bonds between this and the other chain. They do hydrogen bonding, either 2 or 3, depending on. And this one's upside down. Do you remember all that? I mean, if not, hopefully you got notes. You have good notes. You can review it. So you have 2 chains. And then nitrogen space is not connected that way. Oh, it's a problem with the rushing. So again, that's connected to ribose sugar in the opposite direction. This is 3 prime. This is 5 prime. This is 5 prime. Oops, I did it again. This is 5 prime. This is 3 prime. How do I know that? 0, 1, 2, 3, 4, 5. There's 5 prime. This is 3 prime. All right. Here's where there would be an oxygen or not. All right. DNA or RNA for the ribose sugar. We went over this a while back. I'll give you another review on it later in the week. You will need to know some of that. You need to be oriented in that. I'm not going to ask you to draw it out. Is that clear? You don't have to draw it out. But anyways, nitrogenous bases, there it is, kind of simplified. Nitrogenous base, sugar, phosphate, sugar, phosphate, sugar, phosphate. Do you see that? Do you see why we call it a sugar phosphate backbone? DNA and RNA have a sugar phosphate backbone. Do you see that? Sugar, phosphate, back and forth, alternating. And then sticking off to the side is what? A nitrogenous base. And depending on the order of these nitrogenous bases, you have a message, right? How does the information in DNA of the gene get copied into a message in the mRNA? How does the information on DNA get copied in a message in RNA? Through what enzyme? First we know it's G goes with C and A goes with T. Everybody knows that, right?
Yeah.
I can tell you, sitting down with some people during tutoring, that became a problem for some people. Yeah, in RNA. Yeah, see, it says T with A in DNA, but A with U in RNA. It says it right there, right? This little chart you need to know. And I can tell you through tutoring, when I've tutored some kids that have actually come to help long before the test. Notice is last week they were coming for tutoring. And some of you I know are confused because I hear some of your answers and you're not coming for tutoring, and it's really kind of like, okay, we got to get going here. So these pairings, you need to know them, and you need not to get confused. Getting this wrong is going to lead to easy answers that you should know. It's kind of silly to make a mistake on this. So that's base pairing. This is the enzyme. It's actually drawn pretty well. This is RNA polymerase. It's actually more than one protein. It's a complex, but there's a lot more stuff going on. This is obviously simplified. Let's put it that way. This is RNA polymerase. You see the enzyme? Yes, it's this like faded out, see-through little thing here. All right. And it's gonna read. Notice that the DNA has to what? Separate so that you can read it, right? Because when they're together, you can't really read it. And so it's gonna read it. And as it's reading, this is one strand, right? As it's reading one strand, it's going to start, reading, here's the one that's reading. Reading a strand, it's going to read the T, it's going to put an A. It's going to read an A and put a what?
U.
All right? It's reading, and then it's putting in the new strand. It's not replacing. I know that it's easy to say replacing. It makes sense. And you're right, the T is replaced by U in the new mRNA, but the RNA polymerase doesn't it anything. It can only put in a U. It can't put a T there. All right, so it's just going to keep doing that. Why is RNA polymerase a good name for this enzyme? We already went through that. We went through naming it, but go ahead, tell me.
ACE, anything with ACE at the end is an enzyme.
Right.
And then polymer means polymer, and then RNA, so it's like an enzyme that makes polymers with RNA.
That's right.
That matches the,
The what?
that matches
It does match. That's right. It's a better description, but yeah. But this is why what he said is exactly why RNA polymerase is a perfect name, because it's an enzyme ACE that makes a polymer of RNA. Beautiful, straightforward, simple. The name helps you remember what it does. Here again is something you should easily be able to do, which is why I really don't know why it's not. This is all not done already. G with C, C with G, T with A with T. Is that right?
No, that's U.
That's a U. That's correct. That's right. This is a U. And I do make mistakes, but that was on purpose. All right. Are we ready? We good? All right. So here's cytoplasm. Here's RNA polymerase. There's some definitions. If I were looking to do a study guide and I were trying to get ready and I were absent on a day we did a lab, if you were absent on a day where you did a lab because you were on a trip somewhere, would you go to sleep in the class on Monday? I don't get it. If I were absent because I was sick, if I was absent because I was hurt on a basketball court, certainly I would be in dress code. But the other thing I would do is I would make sure to get these vocabulary words straight, and I would make sure that they're on index cards. And I would make sure that I knew what these words meant and what was going on in each of these, because you see they're underlined. The writer of this document and my teacher handed it to me, underlined them, listed the definitions, and then went through and went step by step on what's happening. And the parts that are underlined are probably important. Well, unfortunately, we don't have time to finish this. We'll finish it in the rest of the day, and I'll upload the video. Tomorrow we'll continue reviewing. Good luck. Please don't wait till tomorrow. Please continue to review and ask questions and do work, or you're going to find a hard week next week. Let's look at this. Notice the process of transcription. The process of transcription is similar to DNA replication. You would know that because we haven't gone over DNA replication. That's what you're going to work on over Christmas break. What are some similarities between transcription and DNA replication? Well, you're copying. DNA replication is doing what? First of all, you're using an enzyme. You would not know this, but using an enzyme in both. Two, you're making these are similarities. Using an enzyme in both, you're making a copy. In both, you're making a copy, right, of the message. What are the differences? The differences is, they're using both an enzyme, but the differences is you're making an RNA, transcription versus replication. You're making DNA in one, and the other you're making RNA. Here you're making two copies of the one for each of the nucleotides. Here you're only making one. So those are some differences and some similarities. but we haven't gone over DNA replication, so I don't expect you to focus too much on this. I think that's fair. The whole chromosome is, and here's again, the whole chromosome, both strands are copied. So in DNA replication, the whole chromosome is replicated, and transcription is what? What's transcribed?
DNA.
Specifically, what part of DNA? DNA is transcribed, but what part of the DNA? The gene. The gene is what's transcribed. mRNA is made of mRNA is made. mRNA is single-stranded because RNA is single-stranded. Those were the differences and similarities between DNA and RNA that we had gone over back in August. Again, a good final review. Blank polymerase is the enzyme which carries out transcription. RNA polymerase. Real easy. Again, doesn't seem like a lot of homework to me. T is thymine is replaced by what?
You. We know.
And A is paired with, I'm sorry,
No, the first one is A.
sorry. I misread that. Thymine is replaced by U, right? Which equals uracil and RNA. So A in DNA pairs with U in mRNA. Does that make sense? The T is replaced by U in RNA. So anytime you see an A in DNA, it's going to make a U. It's going to put a U in RNA. Wow. Okay. Well, that one took care of that one. Next page, already on page 6. Very nice, or whatever page this is. Summarize what you've learned. Explain how a gene directs the synthesis of mRNA molecule. Include an explanation of words of phrases, base pairing rules, complementary nucleotide, cytoplasm, DNA, gene, mRNA, nucleotide, nucleus. So you have to use these words. The stuff, in other words, the stuff we said we went over in class for 4 days. So you have in the nucleus, and I'm not going to write the whole thing out, but in the nucleus, you have DNA that has a promoter region. Which attracts what? RNA polymerase. The promoter region comes before the gene. The polymerase reads the gene. At the end, there's a portion of the DNA that's the end. It's called, it has a termination sequence. The gene is what's read. RNA is made. RNA comes out of, let me do a different color here. RNA comes out of the RNA polymerase. It's processed chemically by different enzymes, including, does anybody remember what the enzymes are? Specifically, one enzyme that's going to cut this up and make it into mRNA. Spliceosome. The spliceosome cuts it up and makes an mRNA. It's in last week's work. The mRNA has also a poly A tail.
Thank you.
Go ahead, put it down. Thank you. And you would want to probably describe RNA replaces T in RNA. The RNA polymerase is mentioned. This whole thing's called transcription. Nucleotides, RNA nucleotides are used. You probably want to write down that they're RNA. And then the mRNA goes where? Goes to cytoplasm. and the ribosome. So again, if you found that hard, if it took you a long time to write something like this, that just shows you don't understand the stuff, which is not a problem right now, but it will be a problem on the test. So I think it's time that you start to really focus in learning what the steps are. So now, in the cytoplasm and the ribosome, we're going to do translation. This is what you did in an activity together in class after listening to 20 whatever minute explanation how to do it. You took DNA, you took the gene. This represents the gene, or at least a portion of the gene. Here you have the sequence. That sequence then gets converted to codon sequence, mRNA. This is the mRNA. Every 3 is a codon on the mRNA. What's it called on the tRNA? Well, if this is the codon, what's the tRNA? What's going to match these 3?
The genes.
The anticodon. Anti means not or opposite. Here's a small codon chart. I gave you a bigger one. You have a copy of it that covers all the amino acids. This is the only thing you need for this lab because they give you the mRNA codon and the appropriate amino acid. Of course, you're going to have to figure out the anticodon. What would the anticodon be? By the way, just as a reminder, this is not necessary for the, if you were doing this at home, you would already be done with this page. But I'm going to go ahead and take a little extra time and talk about tRNA. So here you have this tRNA that sticks out, as we discussed last week. You have 3 nucleotides here. This is an RNA. This is tRNA. It has 3 nucleotides. This is what? What do these 3 nucleotides represent? The anticodon. What does anticodon mean? It means not the codon, or opposite of codon. This is the codon, so what's this going to be? If this is the codon, what's this going to be? It's going to be the opposite, right? I heard some people actually answer, which is great. So if it's ACU, then the anticodon would be what? UGA. So you would look for a tRNA, as I explained in that 20-something odd video, that if you paid attention would have helped you answer these questions in a blink of an eye. You had a tRNA, you had 3 spots for letters, and then you had this little spot that said up here that said amino acid. You have to find the amino acid that matched that sequence, and this could be UGA, right? So you'd have UGA and then this word amino acid. You had to cut out the amino acid and paste it there, or not paste it, but associate it with this. And why associated? Because you have to remove the amino acid, right? So here it is. There's a blank space for amino acids. UGA. What is it then? If this is UGA, what's the anticodon? This is the anticodon. What's the codon? I don't see a UGA here, Mr. Mendoza. I don't get it. Why isn't it here? Because this is what? That's what's on the mRNA. This is on what? This is on the tRNA. This little square represents the tRNA. They didn't draw out a big old T. They just put a square. So this is the anticodon. So what am I looking for here? The opposite of the anticodon, right? So if I have UGA, what am I looking for? ACU. And so what is ACU then? Threonine. Or 3 and 9, however you want to pronounce it, I don't care. THR. That's what would go there. Not hard, really. And this is what's going to take it to what? This tRNA is a carrier. It takes the amino acid where? What's going to put the amino acids together? The ribosome. The ribosome. I think you guys have some work to do on getting this together. You have to get this process down. You have to understand the process. The mRNA is meeting up with the ribosome. The tRNA is bringing this amino acid over to the ribosome. What's it doing? It's going to match the sequence up. This is transcription, but in translation, you're taking this codon, the tRNA is going to match the anticodon to it and bring a specific amino acid. Go on and look at this and see if we can see it. Here it is. Here's the ribosome. Again, is there anything to do here? Circling some anticodons. Okay, real hard. Here's transfer RNA. This is what transfer RNA means. tRNA, transfer RNA. tRNA, an amino acid. So what the tRNA does is it grabs an amino acid. It picks a specific amino acid. It's very specific. This tRNA will only grab this one will only grab leucine. It will never grab any other amino acid. There's more than one of them will grab leucine. There'll be another one that grabs leucine as well. But this tRNA only brings leucine to this ribosome. So the codon matches the anticodon. They match, they associate. That association allows the ribosome to bring the two amino acids together. And what happens between the amino acids? They get what? Speak.
Well, I said they'll link up.
They link up. They form a bond, a chemical bond, a peptide bond. They form a chain of amino acids. That's all there is to this. You could have read or you could have listened in class. I don't care how you learned it, but it's been taught. Here it says anticodon explains anticodon and codon for you in writing if you're confused. And then it says what? What does it say to do? Circle the anticodons in the tRNA molecules in the cytoplasm in the figure. Use arrows to indicate where anticodons in tRNA are matched with the complementary codons in mRNA in the ribosome. So you'd circle what? What am I circling? I'm circling the anticodons. There they are. 3 of them. There's the 3 of them. Here's another 3 just in case. And look over here. There's some more over here and over here. Those are the anticodons. Then it's supposed to do what? What are you supposed to do?
Use arrows to indicate where anticodons are matched.
So where are they matched? Here. Right? This is a match. And over here's another match. Right?
Yeah.
I don't know, but that took me. If I didn't have to explain all the details, if we were all understanding what was going on, that would have taken me what, seconds to do? All right. Here is translation modeling procedure. So this is what you were supposed to do. You were supposed to read. Instead of you reading, I took the 20 plus minutes and explained it to you and did it in front of you and sent you a video helping you. So here is threonine. The mRNA says ACU. The anticodon will be UGA. What was this supposed to do? It was supposed to help you identify what amino acid went with which tRNA little square, right? Because you cut out the squares, you cut out the tRNAs, but some people were like, okay, but what amino acid goes with what? This is supposed to help you. So ACU is UGA. What is CAU? GUA. What's the next one? GGA. Sounds like everybody did it. GAC, CUC, and CAC. Doesn't seem like it was very hard. Again, that's it for that page. Oops, that sounds like a lot of homework. No, not. Next page has you taking a look at, as an example, this is what it should look like. No work for you to do there, except actually doing the work that I modeled for you and sent you the video on, that we watched in class. Now, What's the only thing you had to do here is use this. It says what? Use an arrow to indicate the anticodon in the tRNA and use a star to indicate the amino acid. All right, so let's do that quickly. An arrow to indicate the anticodon. Where's the anticodon? CAC. It's what's coming to match the RNA. I think if everybody paid attention, you'd see that the codon is, the mRNA is here. It's this long thing. It goes in and out of the ribosome. It's a chain of nucleic acids bound together, formed in the nucleus. So here you have, there's your anticodon, and it says, and use a star to indicate the amino acid. What amino acid is it? The valine. There's my star. Quite easy, not hard. 4 pages. I think I did 4 little marks on 4 pages. Put a rectangle around each codon. Where are the codons? They're underneath. They actually say codon. Again, not hard. There's one, and there's the other. What's the difference between codon and anticodon? The codon's on the mRNA, the anticodon's on the tRNA, and they're opposites. I don't know, but I just don't see it. I don't see the difficulty nor the extreme amount of time that this takes out of your life. Draw a line to indicate the location where you put the piece of tape to represent the covalent bond between the first 2 amino acids. Draw a line to indicate the location where you put a piece of tape that represents. So this is exactly what I showed you in the video. So I was doing your homework for you. Huh. So where would I put the piece of tape? Between valine and histidine. There's my thick line. That's where you should put the tape. Hopefully you did not tape the amino acid to the tRNA. And that's it for that page. Again, I don't see a lot of work there. Moving on to 10. Here says what your model should look like again. What happened to the first tRNA? Notice there is no tRNA. There's no anticodon here. There's an amino acid. It's taped together, right? This is taped together. It's bonded. What happened to the other tRNA? What happened to the first one? What happened to the first tRNA? It goes to go collect another amino acid. It literally falls off and goes and gets another amino acid. If you read the procedure, they actually explained it to you here. I explained it to you in class, but they explained it to you again in the reading, that as it comes along, the tRNA falls off, and actually in the 20-some-odd minute video, I told you the answer to this. I showed you I literally took the tRNA and said, it goes away. So there it is, the valine. These two are stuck together. tRNA goes and gets another valine, right? Because that tRNA only picks up valine. There's more than one tRNA that picks up valine, but that one only picks up valine. Does that make sense? Am I making sense to you? Yes or no? This tRNA is specifically histidine. There's more than one tRNA that picks up histidine, but this one only picks up histidine. I promise you there's going to be a question on this on the final. So they match up, these bond together. Explain why a cell needs both mRNA and tRNA to synthesize, mRNA and tRNA in order to synthesize a protein. Now I explained this in class. Explain the function of the mRNA. What does mRNA do? That's right. It brings a message from the gene, the DNA, into the cytoplasm. Perfect. Done. What's the tRNA doing? Transferring what? Yes. It's transferring what though? Amino acids. It's transferring the amino acid from the cytoplasm to the correct part in the message, right? So it's the tRNA is bringing the amino acid, and it matches. You need the tRNA because that's what's bringing the correct amino acid. The message is there in mRNA, but it doesn't get translated into a protein unless you have a tRNA that's going to transfer that amino acid over to the ribosome. So however you wrote it, your answer should explain that. You need both, because you can have a message, but if you don't know which amino acid goes with which codon, you need the anticodon for it to match. Does that make sense? So the tRNA must be specific to that particular amino acid, and there must be more than one tRNA for each amino acid, because according to our chart, there's a lot of redundancy.
The next one is like the same question.
Next, explain the function of tRNA and how tRNA and mRNA work together to result in the right amino acid. I just answered that, right? So literally in 4 minutes having a discussion with you, wow, if you could have had that discussion in advisory or during class or at home in a study group, that would have solved your issues. Or you could have read the book, or you could have listened in class or watched a 20-some-odd minute video. Whatever way you wanted to find that answer. I want to reiterate that the answer to this is pretty straightforward, and that's that, you know, with the message is on the mRNA. But the message is just a series of nucleotides. It's just a bunch of nucleotides. There's nothing that from that could possibly get you to an amino acid, right? In order to make the message into an amino acid chain, you need the amino acid, which comes to the ribosome by way of a tRNA. So the tRNA is, again, stands for transfer RNA. So that has the amino acid on it. And the codon, the anticodon is there as well. So you need to have the tRNA. So the tRNA brings, so this is the mRNA. The mRNA is just the message. It's a coded message. The tRNA transfers or brings the, number one, it brings the mRNA. Sorry, the amino acid to the ribosome. And secondly, it decodes the message. It's the key that helps the ribosome decode the message, is what I should say. So how does that do that? It does that with the anticodon. So if you have AUG, then on the anticodon would be UAC. And UAC matches, and the fact that these two match allows the ribosome to order the amino acids in the order that the gene meant it to be. The gene determines this code because mRNA was determined by the gene. There are other factors, as you saw, things are cut out, so there's all kinds of control mechanisms that change, and we've only scratched the surface on all the things that you'll have to learn later. But in essence, you have the DNA, the message in your DNA, which is inherited from father to son, mother to daughter, mother to son, father to daughter, throughout the generations, is that DNA that's secluded and protected in the nucleus, that's wrapped around histones and put into chromosomes. So those packets of genetic information get unwound, read by mRNA polymerase. An mRNA is produced that leaves the nucleus, goes into the cytoplasm, finds a ribosome. The ribosome then connects with the messenger RNA, which has the code that's a copy of the DNA code, the gene in the chromosome. And that is read by the ribosome, and the mRNA codon matches the anticodon, which then translates the message into an amino acid sequence. That answers all those questions there. Proteins in biological organisms include 20 different kinds of amino acids. What is the minimum number of different types of tRNA molecules that must exist in a cell? If you have 20 amino acids, what's the minimum number of tRNAs? 20. One for each kind. It has to be specific, right? So if this tRNA, whatever that tRNA is, picks up histidine and lysine, will this system work? No, it has to be specific, because otherwise the code doesn't mean anything. If I say cat, and every time I say cat, I mean hat, sky, sun, building, human, does the language mean anything? No, it has to have meaning. What gives this language meaning is that the tRNA picks up only a specific amino acid. So you need at least 20 tRNAs. Look at the figure on page 5 and explain why it makes sense to use the word translation to describe protein synthesis. So what's the figure on page 5 look like? Why does it make sense of translation? Because you're taking a message and you're doing what? You're changing it to a whole different thing, right? Instead of taking a message, you're making something. You're translating it into something new. The message is not there anymore. You no longer have ACU. You don't have codons anymore, anticodons. Now you have just an amino acid sequence. So it's a completely different language. All right. Explain why it would not make sense to use the word translation to describe mRNA synthesis. Because, go ahead. Because it's not translating. You're not changing the language. It's still nucleotides, even though you do have a U instead of a T. And yes, you're using RNA instead of DNA. That's true. But it's still a nucleic acid sequence, right? It's a nucleotide sequence. You still have 3-letter codes. It's the same language, just maybe a different dialect. But now when we're taking RNA, the message and changing it into amino acid sequence, now you're changing the whole language. So that's why. What part of translation depends on the base pairing rules? Translation. What depends on the base pairing rules? I'll let you answer that, considering I just went over the whole thing. That's a good answer, but specifically what part of that transfer?
The information from DNA and the gene
No, translation. Translation's happening in the cytoplasm. Yeah, what about it? What two things? That's right, perfect. The codon and anticodon matching. The matching is what depends on the codon base pairing. So here's some more reading. Here's some things that are highlighted. Again, I would consider them important. Different alleles can result in different characteristics. We talked about that. You learned about alleles in middle school. Big A, big T, little t, dominant, recessive. It's time to go. So you'll have to get the rest of this from the other class. Hopefully we have more time in the other class. Hopefully someone's done this so we don't have to go through the codon. If not, if you haven't done it, make sure you have your codon chart, your universal code and your study guide out. So in the table below, and they give you this table, there's 2 genes. There's a normal gene here. It's a normal gene. And then there's the sickle cell gene. The phenotype, what you see in one is a normal red blood cell. So in one you have a nice normal red blood cell. It's beautiful, works perfectly. There's 4 hemoglobin molecules that are perfectly, and they're shaped. And there's in the center, there's this +2 iron atom, ion that carries the oxygen, binds the oxygen. In the sickle cell, the phenotype is this. That's what you see, right? You don't see this gene. You don't see this gene, right? Does that make sense? What you see is either this cell or this cell. If you look under a microscope, you're either going to see this or this. The phenotype is what you see. It could be color, it could be, in this case, cell shape. That shape determines the function. This cell gets stuck in the capillaries, causes strokes and painful blood clots and liver issues and blocks the heart. It could kill you. So it's really not anything that you want to play with. In this sickle cell disease, thank God we have treatments that can help, but it's still a long-term, lifelong issue that people have to deal with. Sickle cell trait does not lead to many issues at all, because it does not, that's because trait, they call it a trait. You have in other words, you have the gene, but it's recessive. You have the dominant form, which is normal. So your cells are pretty normal, even though you still have a gene that's producing a hemoglobin molecule that's not quite right. Yeah.
So my sister, she got the trait. So like, say she found somebody else with the trait, could they make a baby that got?
Of course. And do you remember your Punnett squares? In middle school, did you do Punnett squares? We're going to come back to sickle cell again and again. We'll come back to sickle cell when we do genetics, and we'll come back to sickle cell again when we do evolution. Because there's actually an advantage. Your sister has an advantage. If she goes to places where there's malaria, do you have sickle cell trait? All right, so if you both go traveling and there's malaria in the area, your sister is resistant to malaria. It's harder for her to get malaria. You are not. You could get malaria and you could either die or get sick and have lifelong issues. So it turns out that having the trait, having one copy of the gene, is an advantage. Having two copies gives you the disease. So this is a recessive trait. Okay? Well, let's take a look at this. Transcription produces this. Let's go ahead and transcribe it. This is in what? What is this molecule in? What is this molecule? DNA, correct. How do you know it's DNA? Besides the fact it says gene. Because there's a T. It says gene. If it's a gene, it's a DNA. But it says T. And transcription produces what? RNA. Right? The codons. So we have to, I like to do this. I like to break it up into three, so when I translate or I transcribe them into an mRNA, when I copy the message into an mRNA, I get it right, because I can easily mess it up if I don't. All right. So CAC would be transcribed as what? How would I transcribe this message? G-U-G. And then the next one? C-A-U. Next one?
C-U-G.
C-U-G. Next one?
A-C-U.
A-C-U. Next?
C-C-U.
And last?
G-A-G.
All right, has anybody done the translation? Now wait, what does the translation produce? A protein, an amino acid sequence, a primary structure. All those would be correct answers. What amino acid sequence? Anybody do this? Who knows what the amino acid sequence is? Go ahead, tell me. Yeah, tell me all of them. Go ahead. VAL. Say again. Histidine. HIS. LEU. Go ahead. Huh? Proline? I'm sorry, I can't hear you. Threonine? THR. Huh? Proline, PRO. And glutamic acid, GLU. All right? So that's the amino acid sequence. We've translated it. Now this was the code. It was coded. It was a coded message. It still doesn't make any sense. And now we translate it into language that makes sense, a protein. Now we have a hemoglobin molecule. Now we can take 4 of these and we can make a red blood cell and it will be normal. This makes sense. This is something we can communicate with. This is a code, both the DNA and the mRNA. All right. So let's see what a change. What do you see a change? Is there a change in what is the change between the two? These are two alleles. I gave you the answer, but these are two copies of the same gene. They produce a hemoglobin, both produce a hemoglobin, right? One you got from a mom and the other from a dad, okay? But is there a difference between the two? Are they the same? Is this person homogeneous or is this heterogeneous? Is this two different versions of the gene or are they the same version of the gene?
Different.
What's different about them?
Because
Can't hear you, say again. So this is an A instead of a T. Do you see that? Somebody say no? Okay, so this is a T up here, and let me put a little asterisk by it. And this is an A down here. So there's a change, a fundamental change. How'd that change happen? It was some mutation that happened a long time ago. Mutations happen randomly without any real direct cause. They could happen because of UV radiation, they could happen because of chemicals, they can happen because of other issues. So mutations do happen, but those but whether they're, for whatever the reason is, and we'll talk about it later, whatever the reasons are for the mutation, we can talk about it later. The cause of the mutation being not the point, it changes the DNA, right? So you have one version of the gene here, another gene version here. This version leads to a regular normal cell. This version, if you have two copies of this, it leads to a sickle cell. Well, let's see what's the cause. What happens if we do that? All right, so let's look at it. What is? Let's do it again down here. That's CAC. So everything else is going to be the same. Do you agree?
yes.
So I can put the same, everything else, so this would be GUG, this is CAU, this is CUG. By the way, how'd you get the amino acids? ACU. Anybody know how they got the amino acid?
It's from the chart.
It's from the chart. You get it from the chart. When you got the chart, what did you read? Did you read the anticodon or the codon?
The anticodon.
You read the codon. You use the codon for the chart. The codon, what you see on the mRNA. GUG. So the codon take a look at the codon on the chart for GAG. The difference between GAG and GUG is one letter. It goes from A to U. That's it, that's the change. Instead of A, you have a U. Because of that, all the other amino acids in them, and this protein is actually much longer than this. Obviously, this is done, I hope it's obvious to you that this was done in order to show you that one small change can lead to a big change. We're only focusing on the area where the change occurred. The protein is much bigger. So up here you have glutamic acid. Now, because of this, what do you have? VAL, valine. That change causes sickle cell disease. If you have two proteins, two hemoglobin proteins that produce this valine version of the protein, it doesn't work very well, and the entire cell sickles, changes shape, which causes all kinds of cascade of issues. So that's really an important point. There is another important point which really is not emphasized by this document, but I would like to take a minute and spend time on it because I can tell you that it leads into something called the silent mutation. This mutation is not silent, right? You have a real issue because of the mutation. Stand up. You go somewhere in the room where you can see. Stand up. Go stand somewhere in the room you can see. So this is not a silent mutation. What do you think I mean by silent mutation?
It affects a lot.
That's right. It's not too easy to tell. In fact, you can't tell at all. There's no actual change. So what do I mean by mutation?
It changes.
What changed if there's a mutation? Genes. The DNA changed. The DNA sequence changes, that's a mutation. There's all kinds of mutations, and we'll go over all of them. You need to know all of the different types that we're going to cover, because they will ask it on the end of course exam. So we'll go over the different kinds of mutations. But bottom line, a silent mutation is you change the DNA and nothing happens. All of you have mutations in your cells. Most of you have mutations that no one notices. It's not noticed because it has no impact. All right? So let's talk about what a silent mutation would look like. So this is GAG. That's on the codon, so I can read this, right? Because this is a codon chart. Do you agree? All right, so GAG, which is here, and gives us glutamic acid, right? Let's identify it on the chart. Where is it? G, A, and then G. GAG is here, right? And which is glutamic acid. Okay. Now, what about the other one? Where is it's GUG, right? So there's GUG. G, again, U this time, and G. GUG, and that leads to valine. Now, I'd like to point something out. This is the normal one, right? It gives us glutamic acid. Do you see that if you would have mutated the G, the last letter, instead of this one, if this letter would have mutated, would have went from a C to a T. So this would have been, instead of GAG, it would have been GAA. It wouldn't have had any impact. Because it was glutamic acid, what would it be after the mutation? Glutamic acid. So that's what we call a silent mutation. There's no change in the protein, so it functions normally, even though there's a change in the DNA. That's how your DNA is slightly different, even though both of you are normal human beings. Does that make sense? So you can have these small changes in areas where they don't change the amino acid sequence. That's why remember what we said in one of the earlier questions? Why does tRNA, what's the minimum number of tRNAs that you need for all the amino acids that we use in the body? 20. That makes sense, because again, in order for the code to work, it's a code, right? In order for the code to work, each tRNA can only pick up one amino acid. If it's random, then there's no code. This is all gibberish. You have to have a tRNA that picks up a specific amino acid and brings it to the specific spot on the mRNA. If it's not specific, then it doesn't count, doesn't work. You only need 20. How many do you have? Each of these is an anticodon, so each of these is a what? Where is the anticodon? Where's the anticodon? Of what? The tRNA. So this is the anticodon. So every anticodon is a what? Is a different what? A different tRNA. Every one of these is a different. These are codons, but for every one of these, there has to be a what? A tRNA. For every one of these, there's be a different tRNA. So we only need 20, but we have a lot more, don't we? There's a lot more than 20. Why? Because there's 6 of them that carry leucine. 6 different ones that carry leucine. There's 2 of them that carry phenylalanine, and 4 that carry serine, and 4 that carry proline. You see that, yes? Yeah. So there's redundancy. It's called redundancy. You have different, even though they're specific, even though the anticodon for GCU, that tRNA that matches with GCU, will only carry alanine, even though that's true, there's 4 of them that carry alanine. Why is that? Because this allows for a mutation to happen and not change the primary structure. In other words, we know mutations are going to happen. Mutations happen naturally. So you want to minimize the impact of mutation. You don't want to lose your baby because one DNA nucleotide gets changed on your chromosome, on your baby's chromosome. It doesn't make sense. So life has found a way around this problem. One of the many ways we get around these mutations or these problems with replication or copying the message is we have these buffers, these copies, these extra copies. So if you have a change in this last position, notice the last position is the one that really gives you a lot of protection, then you have that just a little bit added benefit of being able to have a silent mutation, a change that doesn't change anything. Okay? Except the DNA sequence. Yeah? Is it an emergency? Yes?
Yes.
All right, go. All right, so let's take a look at these questions. Did that help you understand what we're talking about? All right. What's the difference between the amino acid sequence of the hemoglobin molecule synthesized by translating the sickle cell versus normal hemoglobin in the RNA molecules? I think we answered that, right? What's the difference? You have one amino acid change. That's it, right? Is that right? Did I answer that correctly?
Yeah.
All right. Each complete hemoglobin protein is more than 100 amino acids, all right? So we only talked about what? Maybe 6 of them? There's actually 100 amino acids in a hemoglobin molecule, and that's for all 4 of them, and there's 4 different ones. And that's 100 amino acids, and that would mean how many nucleotides in the mRNA?
100. Wait. What?
If there's 100 amino acids, how many nucleotides in mRNA?
300.
300. Why 300?
Because it's 3.
Because there's 3 codons per amino acid. And how is the gene bigger than 300 or smaller? If the mRNA is 300 nucleotides long, it's bigger. Why is it bigger? What did you do to that mRNA, that RNA after RNA transcription? That's right. You added the poly A tails, but then you also cut out introns, right? So remember that other sheet I gave you, cut the spliceosomes cutting the mRNA out. So the gene's actually bigger than 300 nucleotides long for one gene. And then there's 4 of these genes, right? Because there's 4 different hemoglobin molecules. So when we talk about this protein is very important then, right? You're dedicating 4 genes on your DNA to this one protein. Of course, carrying your oxygen to your body is important, so that's good. All right, so now let's go ahead and take a look at what happens when the sickle cell happens. This is the genotype. The genotype means what version of the gene you have. Genotype, the type of gene. You either have big A or little a. In this case, it's big S for normal, little s for sickle cell. If you have big S, or big S, little s, by the way, but that's fine. Big S is 2 normal versions of the gene. Then your hemoglobins look like this. They look like almost like a little dance, like a bunch of dancers dancing around in a circle. And you form this beautiful red blood cell that can travel through capillaries, not get stuck, and transfer oxygen normally. Anemia, by the way, anemia means you lack the ability to transport oxygen to the tissues as needed. Right? And the result is that you're weak and tired all the time. Now that could be that red blood cells are dying. Okay? So there's a deficiency or there's something wrong with the red blood cell. It could be because you don't have enough iron, because iron's in the hemoglobin, right? It could be that you're bleeding. Either you have heavy flows because you're a lady and your monthly event occurs and you bleed. You're a person that bleeds a lot, and that happens. That's why ladies can be anemic once a month, and so you should keep an eye on that to your physician. It could be that you're bleeding internally and not know it. For instance, colon cancer can lead to bleeding in the colon, rectum, and that's why it's a good idea to keep looking into the toilet when you're done with your business, because if you see black tarry stool, if you see a change in the color of your stool, you should be concerned because it could be bleeding. So also you could have an iron deficiency. You're not absorbing iron well, you're not eating enough iron. Something could be stopping the iron from working. So in all these cases, the red blood cells aren't working correctly, and so you don't have the ability to transfer oxygen the way you could, so you don't get the oxygen to the cells, so you're not able to make enough ATP, so you're what? Tired. You're not making enough ATP, you don't have enough energy. So you can't do the work that you need to do, you can't focus. So anemia has all those causes. Sickle cell is a type of anemia. So I just listed a bunch of anemic possibilities, right? Low iron is not sickle cell, but you can have anemia and not have sickle cell anemia. Sickle cell anemia is anemia caused by the cell being sickled, which in turn is caused by a gene that had a mutation, which causes a change in the protein. Look at what happens to the hemoglobin molecules. Instead of linking up like this, in this shape, you get this weird shape here, which leads to this shape here in the cell. So the shape of the hemoglobin in the cell changes, so the cell changes. All right, so circle the arrows in the chart that represent transcription. Which ones show us transcription? Transcription plus translation. Which of these arrows show us transcription and translation? I'm going to circle it. Since nobody wants to make a guess, I'll circle. Is that right? Is that transcription and translation?
Can you explain why?
And what about the other arrows? What do you think? I need to hear some thoughts. Where, transcription and translation, where is it? What is transcription and translation?
Transcription is copying word.
Yeah, go ahead. It's copying the code. I'll give you that. But then what? What is transcription? A lot of silence worries me. The finals next week. Transcription, what is it? Right, so you make it into RNA. What are you making in RNA? Well, what are you making the RNA from? DNA. The whole DNA? Everything? The whole molecule?
No, just a gene.
Just the gene. So, So you're saying what now? That the gene, right, gets turned into mRNA. Okay. The gene gets to turn into mRNA. And then what happens? What's the ultimate? Then goes to what? To translation. And what's translation? Making of what?
Oh, changing it to make the amino acids.
All right, and what's in a bunch of amino acids in a chain? What is it? A protein. So when you're going from a gene to a protein, we call it a what?
Translation.
Transcription and translation together. Protein, transcription, translation, right? Protein synthesis. There they are. Genotype. To go from genotype to protein, you need to do transcription and translation. To go from gene to mRNA to amino acid sequence. Are we good? All right. How does your DNA determine whether you develop sickle cell anemia?
You have the trait.
Not the trait.
A mutation.
A mutation in the DNA leads to an mRNA change in the mRNA, which leads to a change in the amino acid sequence, which leads to structural change. You need 2 copies of the gene.
Can you say that one more time?
Yeah. Homogeneous recessive. Right? So you'd have to be homogeneous recessive. You have a change. I'm going to use a triangle means change. This is the Greek letter delta. Change in the gene leads to a change in the mRNA, which leads to change in the amino acid sequence, which leads to change in the function. Specifically, it leads to the sickling of the cell. Are we clear on that? And we're going to see sickle cell all year, the rest of the year long, so make sure you know what you're doing. This process is heavily tested on end of course exam. Transcription and translation, where it happens, what happens, that you will see the universal codon chart, the idea that you can have a silent mutation is more than one kind of mutation. We'll talk more about it after winter break, but you'll have to do that winter packet. It's not going to be easy, and you'll have to read chapter 12. That's going to be your activities over the winter break, reading chapter 12 and doing that winter packet. Now, that being said, you need to know your final's going to be about 40 questions long. It's multiple choice, at the end of which you'll have some activities. I don't think you'll take 2 hours to do the test, so we'll grade your test. We'll go over, make sure there's no mistakes. If there's any mistakes in your grade, you need to email me or text me, email me. Jupiter Grade message me is what I meant. Message me, Jupiter Grade. By Friday.
Well, like this Friday?
No, dear. By next week, Friday. So you're at home and you look at Jupiter and you're like, listen, don't wait till 3:18 to take Jupiter Grade message me. Let's put a time on it. By 12 p.m. Friday. If you don't, that's my deadline for any mistakes that you notice.
We take your test.
And honestly, any old mistakes, any mistakes that that don't have to do with the final, I'm not even going to register because we should have already discussed it. Are we clear? You have this whole week. You have next week, all next week. I'm here every day until 3:18. I should have all the grading done for the week by the end of this week. So there should really be no question. We're wrapping this semester up now. This is a wrap up. Are we clear? So if there's any points of confusion, we're going to keep going. Obviously, now we're going to go ahead and hit the rest of this review. And hopefully everything else is pretty straightforward and it's not an issue. You still got all the videos that I have. You have the Khan Academy videos, which are wonderful if you don't want to use the ones we did in class, because for whatever reason, you can use the Khan Academy videos. There's other videos like the Amoeba Sisters and there's all kinds of things sources you can come up with online. YouTube is amazing. So you don't have to feel stressed. I'm here. We had tutors here from CASE. A bunch of kids were in the back working with them. I know a lot of kids didn't come, and that was disappointing. You have a couple med students back here going through the same stuff. There's absolutely no reason why you guys shouldn't have been here. That's today. Today's Tuesday. I don't know if they come tomorrow or Thursday, but keep an eye out for them if you need extra help. Or me, you can come see me during advisory or after school. Today, obviously, it can't be after school because Tuesdays and Thursdays are no-go. Saturday, I will be here Saturday, but it's not for you guys. It's for the AP Environmental Science. They're having an AP NIMSY Saturday, so it's not open to the school. It's only open to the AP kids, AP Science kids. And that's from all over the district. If you want to volunteer to come and help, I don't know, you can ask Ms. Perez if she needs anybody. Do you have any questions about what's going on with the test, with anything? As far as next semester, I'm not sure. I'm going to send out a survey and ask you guys what you think about about switching over and getting rid of this open-ended deadline business. I felt I'm too close to the problem, so I'm going to let you guys decide how you want to do it. So keep an eye out for that survey if you have strong feelings one way or the other. All right. If not, if nobody responds, nobody has a strong feeling, then I'll go the direction I feel I need to go. All right? All right, then. Let's go on and finish these last few questions here. Why does a cell need to carry out transcription before translation? We answered this many times. Let's see if somebody can answer it. Why do you have to do transcription before you can do translation? Why not just do translation?
Because you need it. You need to get the
You need what? Why do you need to copy DNA? Why can't you just use DNA right to protein? What's the issue? Yeah? I can't hear you. A DNA cannot leave the nucleus. The DNA can't leave the nucleus. That's number one. The ribosomes are not in the nucleus, and the DNA can't leave the nucleus. Second thing is, that's not how the enzyme works. The enzyme reads mRNA. It doesn't read DNA. You can put a ribosome, which is an enzyme made of RNA, you can put that ribosome on DNA, in with DNA, and nothing's going to happen because it can't read it. It can't do anything to the DNA. But you put an mRNA on there, and boom, it happens. Magic happens. All right, and I'm going to show you some videos because we have some time, thank God. All right, so to summarize what you have learned, explain how the gene depicts the synthesis of a protein, directs the synthesis of a protein, include in your explanations the words amino acid, anticodon, cytoplasm, DNA, mRNA, nucleotide, protein, ribosome, RNA, nuclease, polymerase, tRNA, transcription, translation. Hint, you can answer the question on page 3 and 5 for the beginning of your answer in this question. So anybody need me to go through that again? Do you want me to do it again?
Yeah.
Because we've done it a bunch of times. I heard a yes. So I'll do it again. Maybe you can help me. Maybe we can make this more interesting. You can actually help me. So what do we start with? We have a gene. That's right. On the DNA. Let's say a gene in the DNA. It's part of the DNA, also known as a chromosome, when it's all wrapped up. You have a promoter region, you have a termination sequence, and then you need, the gene goes what? Needs a, what enzyme? And let me do something else. Let me do this. Gene goes to RNA, and this is called transcription. And what is the enzyme? That's right, mRNA. Oh, sorry, that's not true. RNA polymerase. So that's important. You have RNA. What happens at it? We're done?
No.
What happens?
You have to go to translation.
Well, before you get to translation, what happens to the RNA? You have an enzyme called a... That's right. Splice. I never spell this right. Let me look this up real quick. Spliceosome. And the spliceosome is just going to cut out the exons, and you have that in that handout that says genetic code and what exactly is it. And there's a little, at the end of it, transcription, translation, DNA has this beautiful graphic, right? And it says spliceosome, and it says, it shows you that you got the exons are put together, introns leave, they leave. And the exons get put together, get spliced together, get taped together. So then you have, and then plus you put a poly A tail on it. Those two things happen, and what you get now is an mRNA. You got rid of the introns, you put the exons together, you add a poly A tail, now you got your mRNA. Okay, great. We're not done yet though, are we?
No.
What do we do next?
Translation.
Okay, where's translation happening? And let's mark this, because I had a lot of empty stares and quietness when I asked what transcription was. Let's remember that's transcription. The mRNA leaves the cell, right? Not cell, I'm sorry, it leaves nucleus. And goes where?
Cytoplasm.
into the cytoplasm /RER, and that finds a ribosome. Which is made of RNA. I'll say again, it's made of RNA. There's 2 RNAs, and maybe I should make sure I write that. 2 times RNA, a big and a small. So the mRNA plus mRNA plus ribosome plus tRNA plus amino acids lets you do what? Translation. And we went through the process of translation. You should be able to, at some level, you should be able to describe it. So translation. So transcription, translation. Now you have the protein that makes you an amino acid sequence. That's where the tRNA, anticodon, codons match up, base pairing. The tRNA goes and collects an amino acid, brings it over to the sequence, matches it. You have to study that. You have to be able to. If you can do that, if you can describe this in general and remember some of those details we just described, then you should be able to do really well on all those questions. And I'm going to tell you this, 40 questions on the final, about 1/3 of them are going to be on transcription and translation. As many as half of those questions, as many as 20 questions will be on transcription and translation. That's how important I think it is. You will have your chart, your universal code. I will give you genes. I will ask you which mRNA is it. I will give you mRNAs. I'll ask you what do you produce. I'll ask you where it happens. Are you writing this stuff down? Because I'm telling you specifically the questions that are coming. What do you have to do? You have to be able to translate and transcribe. You have to know the names of the enzymes. RNA polymerase, spliceosome. That's not hard. Ribosome. Those are the 3 enzymes involved here. You have to be able to explain why do you have so many tRNAs? Why is there more than one tRNA that carries any specific amino acid? Let me pause this because I don't want to use anyone's name. So, anyways, are we clear that, what was the question someone just asked me?
Repeat the last question.
Why are there so many copies, so many versions of tRNAs that carry the same amino acid? What advantage is that? Why is that an advantage? And you can rewind the video when I post it, and if you don't remember. Because we're running out of time is the only reason I say that. Considering that we all are made up of the same 4 nucleotides of our DNA, the same 4 nucleotides in our RNA, and the same 20 amino acids are proteins, why are we so different from each other?
We got different genes. Different DNA genes.
That's right. But listen, the gene leads to an amino acid sequence, a protein, right? Protein synthesis. That protein, that enzyme, can lead in really diverse ways. And then that protein can influence another gene, or it can influence a protein that can then impact another 2 proteins, and so on and so on. So one change in an amino acid could lead to a cascade. This is called a cascade. One change in one nucleotide on one gene can lead to hundreds of changes in your body.
What did you call it again? A cascade?
Cascade.
a cascade. A cascade is like, you know the dominoes? You know, you ever see those dominoes where they line them all up and they push one and then they just make this ridiculously complicated pattern of falling dominoes? That's what happens in your genes. One protein, I like them too. And that vision of that is what you have to think about. If you have one gene change, one protein change, can then lead to hundreds, thousands of changes in your body. It can impact color, it can impact thought. One gene, it's only, Down syndrome is due to one extra chromosome. One out of 23 pairs of chromosome, you have one extra chromosome, bam, you have all these symptoms from Down syndrome. We're not ready to go yet. I still got a couple minutes. I want to show you a video. Hold on. First, transcription. Yeah.
Oh yeah, we gotta upload that.
Watch. Yeah, I would, if you corrected, if you have made some major corrections, yes, please. This is RNA polymerase. The yellow, these are RNA, this RNA polymerase, this is RNA nucleotides, this is mRNA, this is what? This is RNA polymerase, this is mRNA, these are RNA nucleotides. What is this red thing? I'll say again. This is RNA polymerase. This is making mRNA, so it must be reading what? DNA. This double helix looking thing is DNA. Here we go, I'm playing. Shh.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene.
The bundle of factors are assembling at the promoter region. We call them transcription factors.
A gene is simply a length of DNA instructions.
Just watch for a second. It'll take a minute.
The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands.
It happens just that fast.
The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
That's it.